Our final live presentation today. Thank you so much for hanging in there. And you're going to want to make sure that you're really refreshed and really present for our last speaker, Dr. Dan Kalish. So Dr. Kalish, you probably have heard him speak before. He's been doing this for a long time. He's an IFM certified practitioner with over 25 years of clinical experience. And he developed the Kalish method, which really is a fantastic, through the Kalish Institute, fantastic program that teaches practitioners really how to systematize their practice to be a lot more effective with patients, and kind of similar to Jim Laval, make sure you get good results upfront that patients can feel so that they can continue on their health journey going forward. He is the principal investigator in a research study conducted with the Integrative Medicine Department at Mayo Clinic, testing the Kalish method. And he's currently partnering with industry-leading scientist Dr. Richard Lord, developing a curriculum on advanced lab interpretation. So you're going to get a little taste of the basics and the complexities of a really amazing area in functional medicine testing, which is organic acids. So thank you so much, Dr. Kalish, for being here with us today. And I'll let you go ahead and take the floor.
I appreciate it so much. And thank you for hosting this. This is my absolute favorite subject in the world to speak about. And so I'm just delighted to be here. Quick little announcement here. We are starting, the Kalish Institute's doing an amino acids and vitamins boot camp. These are these 2-month intensive deep dive courses. If you like what I'm talking about today and you're interested in learning more about lab interpretation, you can check that out. And you get a 20% discount through Wellevate if you use WELL2021 as a discount code. And that's, I think that'll be our last course for this year. And then next year we'll have a whole bunch of new things coming up. But my biggest passion for the last almost 30 years has been lab testing. And I struggled for 25 years to understand organic acids and seriously questioned them, loved them, hated them. I had an intense relationship with organic acids. And then in 2015, I connected with Dr. Richard Lord, who I'll talk a lot about today. He's a scientist that developed organic acids for our field. And I'll explain all the history and all that in the second part of the talk. How Richard developed it and what that even means and how organic acids are used in other areas of medicine. But these last 6 years, twice a week, working directly with Dr. Lord has been quite an eye-opening experience. And it's literally, he wrote the book on this subject, right? Laboratory Evaluations is the book in this area, and still is really the only dominant book out there that explains how to interpret these kinds of labs. And so being able to talk with him all these years has just been the most rewarding experience of my life. And what I want to do today basically is share the things that Richard has shared with me. And he spent all these last years in his retirement years, right, focusing on training me specifically, really, as the main doctor he's focused on, because he wants me to get out there. And I'm probably at the right age. I'm 57 and old enough to kind of know what I'm doing a little bit, and young enough that I'm going to be out there for another 20 years probably, to really get the message out for what his intention was with these tests and how to interpret them and the profound nature of organic acids. And a lot of this information has been lost in the haze of time. Richard retired a long time ago. Any of the older doctors on the line right now will remember him because we all talked to him when we were being trained back in the day, and really want to kind of bring back that information and update it with all the information that is new and current in organic acids. There's new information coming out every week about this kind of work, right? So, and I divide the course up, this class up into 2 sections. So for the new doctors, who I would define as your first 10 years of practice, the first part of the class, just an introduction to organic acids, why we do it, how we do it, what it means. And then for those of you maybe 20 plus years in practice, the second part of this webinar is a deep dive into how you can use organic acids testing to determine genetic disorders, which is like the coolest thing I've ever encountered in our entire industry. And it's so amazing that I just want to share the concept with you all. And hopefully this will, if you're, advanced and you already know the basics of organic acids, you can maybe check your email for a few minutes, but don't miss the second half because we're going to get into the complicated deep end of the pool here. So learning objectives, reviewing the types of conditions that respond well, which is basically everything. I mean, back in the day when I used to think, oh, I'm going to run organics on this person, but not that person. No, you just basically, you have to run this test on everybody. And the more that you test people, the more you realize that it's just the basic introduction to human physiology is what this lab is presenting, right? And we can look a little bit at the detail with some of the systems that I've tried to develop to just even wrap your mind around what the test is testing for. okay, which is hard. This is a quote from Dr. Lord's book. You can look at that later, read that. Um, and then really at the deepest level, the test is designed to assess metabolic dysfunction, and metabolic dysfunction as it relates to how the diet is affecting your patients and their nutritional status and their genetics. And each organic acids marker is an integration of information that's telling you about how that person's diet has interacted with their genes. And in some of these markers, you can see genetic disorders. In some of these markers, you can see nutritional problems related to diet. And in some of these markers, you can see both in play at the same time. That's really what an organic acids test is to me. And I think these days, the popular thing, right, is that everyone wants a personalized approach to supplementation. Well, this is as personalized as it gets because this looks at your diet and everything that you're eating and how that's controlling things, and it looks at your genes at the same time. We couldn't get more personalized than this. And so you can use it. We're training a lot of primary care doctors now. You can use it as your annual health assessment if you're a primary care doctor, nurse practitioner, or PA. You can use it as a weight loss tool, or just like, why is my mitochondrial system not working tool for depression or anxiety. You can use it for simple patients, complex people. In the days when I learned to do organic acids, it was sort of saved for people who are really sick and you couldn't figure out their case with the other basic testing. But really, I think at this point, we want to elevate organic acids to being a test for everyone. Okay, every new patient you should run this on. And in fact, at this point in my career, I, in good faith, couldn't speak to a new patient without being, having their organic acids in front of me and really looking at the test and digging into all the things that it shows. And if we're going to try to categorize the test, you can look at it in terms of like different body systems. You can look at it in terms of categories like physiological damage. This is just sort of, I think the point of these slides is really this slide and a couple others is just that you need some kind of model or some way to wrap your mind around the test. And I'll show you a few examples and that you can then perhaps create your own. So this is one example I use a lot in the mentorship classes I teach, which is these 4 horsemen. Is there inflammation? Is the person catabolic? Is there insulin resistance? Is there oxidative stress? And so you can see inflammation, catabolic physiology, insulin resistance, and oxidative stress on organic acids. There's brain inflammatory markers on the test, right? You have the catecholamine markers on the test for stress physiology. You have probably the most precious and important markers on this lab in some ways, the ones related to insulin resistance. And then maybe even more important than insulin resistance, if there is something, it's oxidative stress and the glutathione markers. And so there's just a need, though, because there's so much information for us to create some kind of a model. And if you like the models that I present, go for it, use them. Obviously, these are patient education models, right? More than they are doctor education models. But if you order a complex test like this and you need to interpret it and you're working with a patient, you have to have some way to show them what's happening, and you can't explain to them in detail about the pyruvate dehydrogenase complex deficiency disorder that they have, right? Because that's like way too complicated. But most people can wrap their mind around insulin resistance, and that is a great talking point in saying, gosh, it looks like from this lab that your pyruvate enzyme, the pyruvate dehydrogenase enzyme is not working great, and that means that you're insulin resistant. Let's talk about what that means, right? So that's what these models are for. So you can take the complex data on the lab, translate it into an effective supplement program, and at the same time explain that to a patient. So when they walk out the door and they go home and their family says, well, what did the doctor say? They can say, oh, well, I have the beginnings of insulin resistance. And then when they say, what does that mean? They can say, oh, well, let me tell you what that means. Right. And that's really the goal here, is for us to be able to educate patients so that they understand what's happening. And then here are the 4 horsemen, right? Inflammation, you're catabolic, you're insulin resistant, you have oxidative stress. These 4 sort of sweeping problems that affect many people. And then the question is, what's getting damaged as a result of that? Are your mitochondria becoming damaged? You can see that on the lab, really clear as day. Are lipids being damaged? Is the DNA being damaged? There's markers for each one of these areas, right? And that's what we want to think about when we think more about detail about this test. And then in terms of mitochondrial damage, and I thought I would just break this out because we don't have time in this short talk today to go through each one of these issues right here, but let's just kind of hone in on the mitochondrial part, because that's a popular topic right now anyways, and something that's sort of basic that we should know about, right? And the gist of that conversation really is that you can have mitochondria that are damaged from toxin exposure. You can have mitochondria that are damaged from excessive oxidative stress from any source. And how are we going to kind of conceptualize that, explain that to patients, and then how are you going to design protocols to help fix that? That becomes the biggest problem, right? And so if, for example, you have mitochondrial damage on the lab, and you see that in the section, where they're testing for the mitochondrial markers, then you can immediately look at the glutathione marker, for example, and at the detox markers and see, is there a relationship there? And there almost always is. In other words, the lower that the person's glutathione markers go, and the worse shape that their liver detox pathways are in, the more damage you're going to see with the metabolic markers for the mitochondria. And that just kind of makes sense when you think about it, because one of the things that oxidative stress or a lack of glutathione does that's really sort of devastating is interfere with mitochondrial energy production. And the fact that this one test shows you both of these things in real time allows you to get a comprehensive correction where you can say, gosh, your sulfate is low. That means you're low in glutathione. And I can see 5 of these mitochondrial markers are not doing well. I can see 5 mitochondrial enzymes that are not working well. Maybe there's a genetic problem. Maybe it's, nutritionally based. Maybe it's just from oxidative stress. We're kind of leave that as a question mark, right? But that you can then say, I'm going to give you these 3 supplements to get your glutathione regulated well, and these 4 things over here to get your mitochondria functioning well. And let's talk again in 2 or 3 weeks and see how much this is helping, right? And if you have enough of a comprehensive approach like that, you can really knock out some difficult patient cases in, what would I call it? Like a science-based way or a lab-based way without any kind of guessing, very targeted, in a very targeted way. Okay. And so when you look at these tests, and I have an image on this coming up in a second, this little more graphic image that shows this, but we're really looking at enzymes. And this is a little complicated, but I'm going to show you a little bit about this because I think once the light goes on and this kind of clicks, the interpretation of the labs all makes more sense. The relevance of the labs makes more sense. So not only do we see these major detox pathways like phase 1 to phase 2 detox, sulfation, what glutathione is doing, what glycine is doing, all that kind of stuff, plus antioxidant markers on top of that, right? But you're also measuring these specific enzymes. And it's enzymes that are creating the citric acid cycle. It's enzymes that are responsible for glucose metabolism. It's enzymes that are responsible for the burning of fat. So right off the bat, right there, you've got like an entire body of work that you could do with a patient. There's massive, massive, massive amount of work that you could do with somebody, right? Just based on those markers there. Okay. So first of all, the citric acid cycle, number 1, right, is how we're making energy. And then feeding into that, we have the ability to burn carbohydrates and fatty acids. So let's just go on here and see. Now, we have in terms of underlying causes, we have the physiological damage and where is it coming from. And we're not going to get that much into that today, okay? But just to point out that there's more complexity to this, right? So, for example, if you see that there's mitochondrial problems and you see that there's an issue with glutathione, there's more investigating to do, right? And we're not presenting like an entire, complete model of functional medicine here. We're kind of honing in on a narrow area, but just to also realize that acknowledge that there's deep underlying causes that we're not talking about, whether that's GI infections in the microbiome or gluten or toxic metals or toxic chemicals, or the intense emotional social distress that a lot of people are experiencing now, or spiritual disconnection, which is probably the biggest problem facing humanity and always has been, right, for all of humanity. So we're not talking about all that stuff. We're kind of honing in on a narrow lab-based area, but I just want to acknowledge that you've got to investigate these other things too. And you have to be spiritually connected yourself as a doctor. You have to be understanding of the microbiome as a practitioner. You got to deal with metals. I mean, these are things that we're not talking about today, but that are important. Okay. Now, this is like a slide that's just meant to completely intimidate people. And I say that only half jokingly, because this is what we're up against with a typical organic acids test, right? And each one of these markers requires months of study to understand. And there's dozens, if not more than that of them, right? I don't know, depending on what testing company you're using. There's several different really great quality labs out there. This is just from one of the lab companies that I use frequently, but it's just sort of a survey to realize, whoa, what's even going on here? And you can, however, look at these in categories, and then the categories aren't so bad, right? So the categories would be, can you metabolize carbs? Can you make energy in the citric acid cycle? Can you break down fatty acids? Yes or no? How are your B vitamins? That's pretty profound. How's your brain, the neuroendocrine system, right? Stress and neuroinflammatory markers. How's your digestive tract? You eating enough vegetables? You have a yeast overgrowth? Those kinds of questions. And then the detox section, looking at specific detox markers, oxidative damage, methylation, all those things. So again, it's harder to wrap your mind around this when there's dozens and dozens of complex markers that you need to understand, not only what it means when one is high or one is low, but what does it mean when succinate is high in relation to the glutathione markers, because each of these markers means something different in relation to the other markers on the test. It's pretty complex. However, if you think about it, you got to start somewhere, right? And so if you think about it from a more of a systems approach, you're like, okay, what systems are there? There's the neuroendocrine system, detox system, GI system, metabolism system, whatever we're going to call that, a mitochondrial system. Then you can realize, okay, there's problems with the mitochondria, yes or no. Problems with the neuroendocrine, yes or no. Problems with detox, yes or no. And that's easier to wrap your mind around. And I find that in the early years of doing this, which again, to me, is like your first 10 years of doing this, it's enough to start with a broad overview. And then later on, as you get more and more advanced and more into this, if you want to spend 2 or 3 months understanding that succinate marker over there, which is perhaps a fascinating and important thing, then you can do that, right? But you don't have to understand each marker to its full depth to get started with this work. Okay, and then I also have this little graphic here on the supplements that are associated. And this is a very partial image here in terms of markers. A sort of quick summary, really. There's probably double this. But just again, to wrap your mind around, there is a finite number of potential supplements that are involved and a very finite number of lab markers that are involved. So in some ways, it's not as bad as it seems. And in some ways, it's a lot of information, but don't get frustrated. I've been doing organic acids for almost 30 years, and it gets better and more interesting, and I get better at it every year. I can honestly say that right now I can interpret these tests better than I could a year and a half ago, dramatically better, because I've learned more about the physiology behind each one of these markers and more about the pathways and how all this kind of connects and works together. Now, we're in the introductory part of this talk, right? So that we don't think the basic things that you can't get more basic than macronutrients. And what's the flow of the macronutrients through the pathways? Because there's only 3 of them, right? Amino acids, fatty acids, and carbohydrates. And if you get those 3 nailed and the person's doing well with their amino acids, well with fatty acids, and their carbohydrate metabolism's working also, you're going to have a pretty healthy person. So you can break it out again to these really overly simplistic but helpful constructs, right? And again, you can see on the lab if there's an amino acid, fatty acid, or carbohydrate metabolism problem. Shows up right there on the testing. And I'll show you that right here real quick because that's not intuitively obvious. Here, under carbohydrate metabolism markers, pyruvate, lactate, those kinds of things, or pyruvic acid, lactic acid, the energy production markers there. Here we go. Fatty acid markers, right? There's usually 2 to 4 of those markers, depending on what lab company that you use. And B complex is really, whenever you see a B vitamin marker, most of the time those are derived from amino acids. So they're really talking about amino acid metabolism when they talk about B vitamins. So right off the bat, you can learn from your patient. If the B vitamins are screwed up, they've got an amino acid metabolism problem. If the beta oxidation or fatty acid markers are messed up, they're not burning fat well. And if they have pyruvic acid, lactic acid problems, carb metabolism's off or they're insulin resistant, right? And so these 3 general categories can just give you enough information for a very complex program for a lot of patients. And the solutions are pretty simple. If you see that the B vitamins and amino acid markers are off related to the citric acid cycle or to the B vitamins, just give them free form amino acids. You don't have to nail down which one? We went through this in class a few days ago because this one student is like uber smart. And I think it was fumarate. Anyways, it wasn't like we were arguing, but the student in the class that I was teaching, I was supposed to be the teacher, but the student knew a lot about this too, right? He was like, well, doesn't tyrosine and phenylalanine enter into the citric acid cycle at that pathway? And was like, yeah, they do. Should I just give those? I was like, yeah, you could. That's really advanced thinking, because I was trying to just get everybody in the class to just give free-form amino acids as a general solution. So you can do general solutions and they work incredibly well. If you want to get to the point where you know each individual marker so well that you know exactly what would be the best amino acid for that little particular problem, you can do that too. But you can start generally and get amazing results. And in fact, I've seen this arc with a lot of the doctors. There's, I'm in the mid range, right? I'm in my late 50s. But there's doctors that I've known since I was in my 30s that are now in their 70s and 80s. So they've been practicing for like, I don't know, 50 years at least, right? And there's an arc that I've seen, in those first 10 years of practice, you're learning a lot and you're integrating a lot and you're doing all kinds of new things and it's really exciting. And then you tend to get things more and more complicated because you're learning more and you want it to be more complicated because it's more intriguing, right? And you're helping more people. And then somewhere around the 25 to 35-year mark of doing this, you start to see people turn and they start to say really simple things, like really simple stuff. And you're like, well, that was really profound. And then you go home and you look at your notes, you're like, well, wait a minute, what that person just said was like really simple. And so the people that with more experience than I have, the 40 and 50-year people, they do really simple programs oftentimes because they've been through this whole arc of a career and they've seen that the truth is simple and that a simple free-form amino acid product can sometimes help so many people. You don't need to make it more complex. So if you're relatively new, and you're to this testing, the simple approach that you would take in the beginning can often be the best approach. And it may be very similar to what a more complicated doctor, a more experienced doctor would do after 40 years. Okay. Now, I put together this slide just to talk about what I see in practice like pretty much every day. So these are the pathways that you want to study over the next few years, really understand, and the markers that you should just know inside and out. Beta oxidation, why? Because fatty acids are the main source of energy for the human body. When the human body's at rest, not exercising, but when we're at rest, we're burning 80+% of our energy from fatty acids. The heart uses the majority of it, gets the majority of its energy from fatty acids 24/7, even when you're exercising. Is very, very important. If beta oxidation is not working well, you're going to have a major mitochondrial problem downstream, right? Because you're not going to have enough substrate to make energy from. Glycolysis, insulin resistance is all around us, as we all know, right? And so looking at those markers, pyruvic acid, lactic acid, that relate to glycolysis, you just got to check those on everybody. And if they're off, you got to fix them. That's what I was saying earlier. Like, I can't imagine talking to a patient and not knowing the status of their pyruvate. Like, it just seems like you have to know that. Can you burn carbohydrates or not? Is your glucose metabolism working or not? That seems like a fundamental question. Neurotransmission, I see this all the time. I'm sure you do too. Anxiety and depression, those are well demarcated on the lab. Methylation defects, which is all the rage right now. We see this a lot as well all the time. Mitochondrial problems, oxidative stress. And then I'm teaching this cardiometabolic boot camp right now, and I prepared for it for a couple years. And then when you look at the organic acids test, I mean, almost the entire panel is kind of cardiometabolic related when you think about it, right? Because it's oxidative stress, it's glutathione, it's methylation, it's everything that we think about in terms of protecting your LDLs so they don't get oxidized. Burning carbohydrates properly, insulin resistance, right? So in a way, if you're interested in doing cardiometabolic programs or preventive heart disease programs, maybe cure your if you have a problem with like an insulin related problem, but you're not diabetic, but you want to, prevent the diabetes from happening, right? All these kind of preventive programs that are in the cardiometabolic range, this can be really great, okay, as a test. And here are the basic pathways for the first portion of what we're talking about. And again, on a simple level, I want to look at this for a second. So you have coming in here the macronutrients, fats, carbs, and proteins. You can see at the top. we can measure the body's ability to burn fat through these markers that relate to beta oxidation. It's a very interesting process. It's dominated by riboflavin, which is vitamin B2, and carnitine. And if you're low in carnitine, this whole process doesn't work well. And similarly, if the enzymes that help you process fatty acids aren't working well, there's something wrong with the enzymes, then you're going to have a problem with beta oxidation. Okay, and every enzyme in the body, as you probably know from school a million years ago, is made up of 20 different amino acids. Every enzyme in the body is made up of 20 different amino acids. And if you're low in more than one or more of those amino acids, you're not going to make that enzyme very well. So amino acids end up being really important, not just for running the citric acid cycle, not just as an energy source, but because they're how we construct these enzymes that we're talking about. Okay? And here you can see carbohydrate metabolism, pyruvate and lactate, sometimes also represented as pyruvic acid and lactic acid. Those are the keys. Those markers being normal are the key to showing you that the person is able to break down glucose properly. And if those markers are off, then the person has a problem. Okay, and then similarly with protein, you can see these B vitamin markers. And then if you look in the citric acid cycle itself, they're measuring citrate, for example, cisaconate, succinate, fumarate, malate, right? Each of these markers is measured, and each of these markers, if it's high, shows you that there's an enzyme that's not working right. So if succinate is high, that shows that the enzyme succinate dehydrogenase isn't working properly. And then there's certain nutrients that'll get that enzyme to work right. You just look up those nutrients, it's B vitamins, etc., and give those, prescribe those, and then the enzyme will start to work properly. And you'll start to get the proper flow of energy through the citric acid cycle. And so again, you can design super simple programs with one supplement per section if you're just getting started with this. So if you see a mitochondrial problem, give them a generic mitochondrial supplement. And every company has these. It'll always be called Mito something, Mito Boost, Mito Energy, Mito this, Mito that, right? Give them the combination product that has all the mitochondrial support in it. Crank the dosages up till you get a therapeutic response that's reasonable. Same thing if you see a problem with the blood sugar related markers. Give them a combination product that has all the blood sugar support in it. So you don't have to get complex. You don't have to figure out, I mean, this is what we do in the mentorship. I think this is fun, but you don't have to do this, right? You don't have to figure out how much niacin does that person need and how much thiamine does that person need. That is totally unnecessary. You can do really simple programs and get amazing results. Okay. Same with neurotransmitter protocols. Every company, the big companies that we work with, the popular ones that are professional only, right, will have a combination neurotransmitter balancing supplement. So if you see something wrong with the neurotransmitter section, you can keep it as simple as that, just giving the one product and calling it a day from there. Okay. Okay, now that's the basic explanation of organic acids. If you're getting started and you want to just do some testing, you can keep it simple and prescribe one-off products for different categories. Now I want to talk about the advanced concepts and really what's happening. So in the late 1960s, a rather young Dr. Richard Lord went to the University of Texas at Austin to get his PhD in biochemistry. And at that time, one of his professors in his PhD department, uh, in the biochemistry department, was a man named Dr. Roger Williams. And I have this, kind of like, after the fact, glossy dream of how this happened. But Richard really did talk to this guy every day. But and Roger Williams really did discover pantothenic acid. He also discovered vitamin B6. He was one of the main people that discovered folate. So this was like a hotbed of nutritional research. And I just have this image of Roger Williams sticking his head out of the this old door in the University of Texas Austin saying, "Hey, Richard, come down here. I think I found pantothenic acid." What an amazing time, right? And so at that same time, organic acids testing was also being done. Okay. And one of the things that was happening then was that organic acids testing were being developed to check for inborn errors of metabolism of newborn babies. And so Richard saw both of these things simultaneously, and he's a very smart man, like at the genius level. So he saw, oh, well, Dr. Williams has just discovered pantothenic acid and B6, and Maybe folate's kind of important for human survival too. And there's these other doctors over here doing organic acids testing, and they're finding that these newborn babies have genetic disorders that are life-threatening. And he thought, well, couldn't we put these two things together? In the late 1960s, Richard thought, wait a minute, maybe we should be testing organic acids, not for the newborn babies. Of course, we have to screen for them. We don't want a newborn child to die from some, very rare genetic disorder. But what about all the adults that have the same genes, but they're just a much milder expression, not the full-blown disorder, but just a very small version of that disorder. And so he got a chance to do this a few decades later when he teamed up with Andy Brawley, and they started to do the first organic acids testing. They took the same exact lab tests that are used in conventional medicine for determining whether a child has a life-threatening disorder, right? And they just created adult reference ranges for them. That was it. That was the whole thing. And there's a ton of papers on this. Like you could read about this literally forever. But that's the origin of organic acids testing. They were originally designed for newborn babies to detect whether there was a life-threatening genetic disorder, and then they were applied with different reference ranges to adults to look for nutritional disorders. However, and this is a pretty big however, what got missed in there is that you can still use the adult reference ranges to look for genetic disorders. That's the origin of this test, and it has applications for us that are pretty profound. So here's a little quote from that paper I just showed. Inborn errors of metabolism affecting enzymes or transport proteins required for catabolism of amino acids, lipids, or carbohydrates, what we're talking about, the macronutrients, lead to pathologic buildup of upstream substrate metabolites. That's what these genetic disorders are. That's actually exactly what we're looking at on an organic acids test. Okay, and there's whole groups. There's this group called the Society uh, for Inborn Errors of Metabolism, right here, and they have this adult wing that I actually joined last year, Adult Metabolism Physicians Group. It's not the easiest group to join, but and it's not very expensive. It's like $100, but it took a little while. And that group of doctors put together about 10 or 12 years ago this book, which is quite interesting. Highly recommend you all getting it if you're interested in this kind of workup. Inherited Metabolic Disease in Adults, a clinician's view, and a clinical guide. And this book goes through what's happening in that world, right? Which is that these tests that were originally designed for and still used for newborn babies for screening like diseases like PKU or urea cycle disorders that are potentially life-threatening can also be applied to adults, and that there are inherited metabolic diseases in adults. But when you look at the overall community of scientists studying inborn errors of metabolism, the vast majority of them are looking at newborn babies and childhood disorders. But there's a small, really dedicated group that's starting to look for the same problems in adults. Does that seem similar to something that Richard Lord was thinking in the late 60s? It's not just similar, it's identical. Okay, so there's now a recognition in the sort of advanced neurological / genetics community that these disorders that they see in newborn babies can be can appear in adults. And there's just a couple articles you could read about this literally for years, and I have been. So I just pulled like 2 or 3 things so you can just see what I'm talking about. Think Metabolic in Adults with Diagnostics Challenges. And again, this is the Journal of Neurology Clinical Practice. Neurologists should consider the possibility of an inherited metabolic disorder in adults with neurologic symptoms that may or may not mimic those seen in affected children, such as the case of biotinase deficiency. Biotin-dase deficiency, the enzyme, right, that's related to biotin. That's exactly what we're looking at on organic acids. Now, the problem is that you could have a simple biotin deficiency, and you can see the organic acids that we do positive for the biotin marker. But there are reference ranges that will allow you to see if there's potentially a genetic disorder with that same exact marker. And that's kind of like the news flash that I think is so fascinating. And so when I first met Richard, he told me this, like within the first week of us having our regular talks. And I was just incredulous. I just thought, I mean, I didn't say this, but what I was thinking was, are you telling me I've been looking at these tests for 25 years and no one ever told me that this is like the most profound genetic screening tool? And he's like, well, yeah, Dan, that's why I developed them. It's kind of like, well, then I was thinking, well, why, Richard, didn't you ever tell anybody? And so it just got lost in the haze of functional medicine. And what happened was that organic acids were kind of watered down to be a really good nutritional screening tool, but their origins in the 1960s of Richard's mind of them being this genetic screening tool and how this was a new tool that he could use to see genetic disorders in adults playing out just never got taught in a comprehensive way. And so when he and I started to have further discussions, 6 years ago, I was like, you've got to come back in action. Like, this is like not okay that you're retired and I know you're living by this lake and it's beautiful. You got your kayak, but look, we need some help out here in the field. So he very graciously agreed to come back. He has rewritten all of his books. He has done hundreds of hours of recordings for me at the Kalish Institute. We have had thousands of hours of discussions. We talk twice a week for hours and hours each time about all these subjects. And I'm really here now trying to reach out to other doctors to say, hey, this is really amazing stuff. If you're doing organic acids, you've been in practice for 20 years or more, this is the deep end of how to interpret these. And you can just run with this in a really profound way. And so you can use the test as a simple nutritional tool and give simple supplements, or you can get into this level of it. All right. And I just want to show you a few simple things here. This is on mitochondrial disorders, and then I've got a little bit on glycolysis, and then urea cycle, and then we'll have some questions. So here's a map. This is from a research article on organic acids and genetic disorders of all the different potential enzymes that could have a genetic crack in them, right, a SNP, so that that enzyme is not working properly and you could have a mitochondrial disorder. And now, I mean, the type is so small, I'm not expecting you to read this or memorize this or anything. I can't even really see it. I need my glasses to even look at it. But the point of this is that there's a lot of them, okay? And your mitochondria is just riddled with potential enzyme defects that can cause mitochondrial problems, and that you can see all of these on organic acids testing. You can see the nutritional deficiencies, and if you learn how to interpret the reference ranges properly, you can see the genetic disorders as well. Doesn't tell you what gene is operating, but it tells you the functional expression of that gene. And I think an easy way to really see this clearly is one that I see in practice all the time. It's probably one of the biggest issues that we all face with all our patients all the time, which is insulin resistance, right? And so here you can see a glucose molecule coming in. It's getting split up through this process we call glycolysis into two pyruvates. That's happening out in the cytosol. Then those pyruvates are sneaking their little way into the mitochondria, and then there's an enzyme, right, that's responsible for all this. And then the pyruvate's going to turn into acetyl-CoA, and then we're going to make energy out of it. So the enzyme that makes this all possible is called pyruvate dehydrogenase. Sometimes it's referred to as the pyruvate dehydrogenase complex. And it is highly susceptible to not being structured properly, right, because of some kind of SNP. And so you will often see people who have a genetic tendency to have very high pyruvate, in which case they're going to be prone to having glucose metabolism problems and more likely to be insulin resistant. And in those people, you'll see sky high levels of pyruvate, which can be solved with, as we all know, right, thiamine, niacin, pantothenic acid, and lipoic acid. Maybe some CoQ10 if you're like feeling really good about things, right? So you can get this enzyme to work properly if you give the right nutrients, and you can see based on the lab if that marker is high. And you can also see based on the lab with the sort of enhanced reference ranges, we might call them, whether there's an actual defect in the enzyme. Okay, and here's what that enzyme looks like. It's rather large and complex. These are all the different amino acids strung together, then wrapped together, right? And so there's many different ways that it can go bad. There's many different structural defects that it can have. Now another, and I just picked a couple of examples here just to show you the general idea. There's also the urea cycle, right? And within the urea cycle, there's all these different amino acids that are converting. There's all these different enzymes. You can see the enzymes down the lower left-hand corner of the slide there. And so when you see something with that many enzymes, each one of these enzymes could have a genetic defect in them, right? There could be SNPs. And I think there's like 300 or 24, I don't know, 350, somewhere around there. There's 300 to 400 different SNPs that can get screwed up, that can screw up your urea cycle. So a lot of potential problems can happen here. So you see problems with the urea cycle a lot that are genetically based. And on a test, pretty simple, you're going to see high orotate, or you might see really low arginine, or you might see low citrulline or high citrulline. There's a variety of different things that you can see that could be related to exposure and nutritional status, or it could be related to genes. And here's another schematic of that. You can see the urea cycle goes through the mitochondria, which is kind of interesting. I don't know. It's kind of an aside. And then again, in the scientific literature now, the modern day, right now, in the last 10 or 15 years, practitioners that used to always look at these pediatric disorders in newborns are now realizing, wait, urea cycle disorders, a life-threatening yet treatable cause of metabolic problems in adults. Okay, and that's exactly what we can learn from organic acids testing. Maybe it's not life-threatening. I've never had a life-threatening urea cycle disorder case in my practice, but I've had a lot of life-ruining ones, right? So again, there's a spectrum here from newborn baby with their problem that's life-threatening to late onset adult disorder with a life-threatening problem to a midlife level onset of a non-life-threatening problem, right? It's the same genetic disorders, but they're just not expressing so severely. And we can see these on the labs. And if you want to get to this intense level of complexity, you can look at all these different ways. And I'm just showing, I just think this is kind of vaguely interesting. But you can, people take this to a ridiculous degree. So you can see each one of the enzymes, and these scientists have broken down what it means in terms of the amino acid levels. So if you want to figure out exactly what enzyme's defective and all that kind of stuff, you can do it. It's not necessary at all. If you just know that there's a urea cycle disorder, give them some arginine and citrulline and B vitamins, and watch them get better. Okay, can be pretty simple treatment-wise. But the idea here is that we also understand there's some complexity behind understanding the genetic knowledge, I guess you would say, that's given to us from doing organic acids testing. Okay. This is a pause for me to remember to drink a glass of water. All right. And then a couple of protocols, because I showed, the idea of, simple protocols, and then here's some more advanced ones. So if you do some organic acids testing and you see a variety of mitochondrial markers not doing well, and I just put a few there, citrate, malate, isocitrate, there's more, there's probably a dozen of them, right, or more. Then here's a standard protocol, just so you can get a sense of what a comprehensive mitochondrial program would look like. 100 milligrams or more of CoQ10 twice a day, could do 100, 200 twice a day, could go even higher than that. Magnesium, again, 200 twice a day, you could go higher if the person doesn't have any loose stool. A B complex to make all this work. The synergistic effect of the B vitamins is critical. Free-form amino acids cover so many things, one or more teaspoons a couple times a day between meals, one of the powder form. And again, you can use combination products, but if you want to get into really specific programs, you can do this kind of a thing, right? And that's not an overwhelming number of products. Most people can handle that, and they can make a profound difference in terms of how people feel. And let's see, we've got one more protocol I put together. Here's just a general insulin resistance protocol again, because these are ones that I do in my practice every day, so I share them. Lab findings are either high pyruvate perhaps or high lactic acid. One of those markers is off, and you're thinking, okay, I really got to get them under control here. You do this program, you retest in 3 to 6 months, you give them the B vitamins, a hefty dose of lipoic acid, magnesium, chromium, biotin. You can use a combination product if you want, to keep it simple, but we're really also saying, this is in general what a complicated program would look like that would be fine-tuned based on the labs. And most patients can wrap their minds around doing this, okay? So I'm going to open it up for questions in 1 minute. I just want to remind you all, I have this amino acids boot camp coming up. And if you're interested, you can contact Kalish Institute. We can tell you about that. And then let's look at any questions that may have come in.
Awesome. Thank you so much, Dr. Kalish. And I can totally tell that this area is something that is completely a lifelong learning as far as how to really get the most out of this type of testing. So, a couple of questions, and this is not CME, so you're welcome to share anything you
Okay.
want. Are there specific lab panels or companies that you like to utilize for organic acid testing? And are there pros and cons to different ones?
Yeah, I mean, I'm totally loyal to Richard all these years. And so the lab panel that he invented is now run by Genova Lab. So I'm just 100% in Genova's camp because all my training's been from Richard, and he's the one that developed that one.
Great. Thank
Yeah.
you. Yeah. And the other question I have is that when we run organic acid testing, the companies put out pretty extensive panels and guides that kind of guide people and instruct practitioners on what's happening. How helpful are those, and how aligned are they with what you've talked about today? And what are the kind of next steps beyond just utilizing that kind of algorithm-based interpretation to really apply your own expertise to kind of bring it to the next level? I think a lot of practitioners are probably at that level, like getting comfortable with reading and interpreting those reports at a basic level and wanting to really take it to the next step.
Yeah, I mean, first of all, I know and I care a lot about the people that work at these labs, and they're good human beings, and the algorithms are as good as they can make them, right?
Mm-hmm.
But that just shows the limitation of a computer. And so I'm very anti-algorithm because it's like there's just too many variables. Like, I don't know. I mean, if your car's brakes failed and you lived in like the mountains, would you like bring it to a shop that analyzed cars by computer, or would you rather be happier if someone got under there and said, "Oh, the rotor's broken, I'm going to replace it." Like when it's this important, people's lives are on the line. You got to dig in there because you don't know if an individual, and the lab can't tell if an individual marker is screwed up because the person eats poorly, or because their mother-in-law is visiting and they're stressed out of their minds, or because they have a genetic disorder. It just can't give you that level of information. But you can ask somebody, "Oh, well, what about your family history? Oh, you have 15 diabetics on both sides of the family. Maybe there's a genetic problem with pyruvate." So I totally support the labs trying to do algorithms, but I think that they're misleading at best, and that you have to dive in and understand that particular patient and understand the pathways on your own. You have to have your own algorithms. That's what
Yeah.
Richard's really taught me.
Yeah, and I love the reports that the labs put out. I think it's a really foundational teaching tool for the practitioner and the patient when they're getting started. And like you said, there's a lot that you can't see in an algorithm. And that's not just with organic acid testing, that's everything. I mean, we talked about that today with like urine metabolites for hormones too. If you have, high free cortisol and low metabolites, there's a lot of things that could be the cause of that. And you, as a practitioner, need to overlay your knowledge to
Yeah.
dive in to understand the results in context with the patient. So thank
But
you so
yeah,
much. And I
but
think this is,
I want to contradict myself briefly on that, okay? And say that because the whole first half of my talk was saying that that's not true. The whole first half of my talk was saying, look, just run the test, get a general picture of what's wrong, and use a general supplement. That's like a simple algorithm, right? Because we want people getting started in the testing, right? So that's the first 10 years is that. And then this after that should be this more complicated stuff.
yeah. And the more tests you run and the more results you interpret, the more information you're going to glean and be able to apply. You just need that
Yep,
experience. So you got to start somewhere and bite the bullet and dive in. So.
exactly.
Great. Well, thank you so much. Any final words of wisdom to practitioners getting started with organic acids? Just start simple and utilize the combo formulas to address the.
Yeah, honestly, you can just start simple. Use the computer algorithms for the first few years, right? Because they're good enough. And then use combination products that correlate with what the computer's telling you. And then individually over the years, start to study each marker so you can piece together your own algorithms ultimately.
Yeah, wonderful.
Yep.
Well, thank you so much, Dr. Kalish, for being with us today and kind of being the anchor to the end of the day. I know a lot of people have hung in because they specifically wanted to hear this lecture. So we're really grateful for you participating with us.
Appreciate it. Thank you very much.
Thank you so much. So as we close today, we've had a big, big day of presentations. If you remember, starting with Dr. Hyman and covering everything from chem panels to Dutch testing to GI testing to organic acids and everything in between. It's been a really amazing day, and I have an entire notebook full of clinical pearls that I'm going to have to translate and put into my own processes and systems. So I want to again give a big thank you first to the sponsors for today's event. We are so lucky to have so many supportive labs as we launch Elevate Labs, really that are excited to be able to provide access to their labs to each of you and provide education in partnership with us. So thank you so much to all of our lab sponsors. Also want to thank the Emerson Elevate team who's done a knockout job putting on this event. And just to give you a couple of updates, one, when you drop out of this webinar, you're going to be landed on a page that gives you a couple of additional options. We have our Wellevate concierge team standing by, and we have created a couple of breakout rooms. So if you want help getting signed up for Wellevate or getting started with Wellevate Labs, choose the breakout room that's most appropriate to you, and you can get small group access to our team so they can guide you through next steps and answer your questions. If you want copies of the slides or you want to be able to view the presentations after the event, you should receive an email very shortly if you haven't already with Dr. Hyman's presentation, which is only available for 48 hours after he presented it. So that means it will come down Monday morning. But you can access that through that link in your email. And then the rest of our presentations will be posted live around the end of this week, along with slide decks. So I hope you guys had a really wonderful time. And thank you again to our fantastic speakers. You guys have a great day.